let's move to the next speaker, Dr. Yanhong Zhang from the South China Institute of Oceanography. So Dr. Zhang obtained her PhD degree in 2012 from Tsinghua University, which actually where I am based right now. And she now is an associate professor in the key lab of tropical marine bioresources and ecology, South China Institute of Oceanography. Her research interests focus on the physiology, ecology, evolution of marine animals, in particular the comparative genomics and environmental adaptation, evolutionary ecology of syngnathids. Today, she will present the study of genomics of monadal pipefish, which will reveal the shared genomic change in syngnathids and convergent evolution with placental mammals. Let's welcome Dr. Zhang.
First of all, thanks Dr. Zhang's introduction. Good afternoon, everyone. It's my honor to take part in this meeting. Today, my topic is about comparative genomics, reveal shared genomic changes in syngnathids and the signature of convergent evolution with placental mammals. My speech contains 4 sections. First of all, you may be asked, why do you focus on syngnathids? Because syngnathids have widely diversity in morphological and colorful. They are the perfect models for what Darwin considered in the origins, and less forms are most beautiful. Syngnathids including seahorse, pipefish, and sea dragon, which are morphological unique group, which comprise 57 genera and more than 300 species. Their specialized morphological, including the teethless tuber mouse and which absence of pelvic fin and scales. And their specialized brood pouch. They are famous for their special reproductive behaviors, male pregnancy. The brood pouch varied from simple gluing of eggs to a pinch of the skin to a semi-enclosed brood pouch and a highly specialized closed brood pouch. A previous study divide the syngnathids into subfamilies. One is syngnathidae and the other is nervinae. The syngnathids are two brooders and the nervinae are twin brooders. The classification schemes implement independent regions of the complex male brooding structure. For example, the monadal pipefish belong to the nervinae, and the tactile seahorse belong to syngnathidae. They have some common common phenotypes and some difference, such as the monadal pipefish has candle fin, but the seahorse loss of the candle fin. In order to find the genetic base of the specialized syngnathids, previously 3 members of the syngnathids were sequenced. They are tactile seahorse and land seahorse and Gulf pipefish. In our studies, we have sequenced the monadal pipefish. The genomic of monadal pipefish was sequenced will be valuable to understand the genetic basis of their common phenotypes and some lineage specialized features. So based on the genomic data, let's first to see the shared genomic changes. Comparative genomic analysis reveals that the molecular evolution rate in the synthesis was higher than that in other fishes. In other hand, we can say the molecular evolutionary rate in synthesis is higher than that in neophene. This is maybe related to the great genomic morphological diversity. Synthesis possessed two sick SCPP genes and only one PQ-rich SCPP genes. In mammals, SCPP genes play an important role in the teeth development. So the loss of all but one PQ SCPP genes may be in the synthesis maybe suggest a rule for the loss of genes and the loss of genes in Dalton and anomaly formation. The pelvic fin of fish and the headlamp are homologous organs, but the synthesis lost the pelvic fin. Previous studies show some associations of dominant genes in pelvic fin and headlamp, such as PTX1 and TBX4. These genes play an important role in the pelvic fin development. So how about these genes and the synthesis? This plot analysis shows that 3 members of the synthesis lost the genes TBX4. So this result will be suggest that the loss of TBX4 genes is common features in the synthesis because the 4 members include synthesis and a member of neophene. Furthermore, we do the knockout experiment. Knockout of the TBX4 in zebrafish, we can say the mount zebrafish show a pelvic fin loss phenotype. This phenotype is similar to that of the synthesis. And in the mount zebrafish, there result any other changes in the morphology, such as there hasn't any change in the pectoral or the medial fins. So the loss of TBX4 genes may be related to the loss of pelvic fin in synthesis. Besides the genes loss in the synthesis, we also focus on the genes and the genes expression, such as the protocanterin. Protocanterin is which belong to the canterin superfamily. Based on the functioning domain, these genes can divide into cluster groups and non-cluster groups. They have different functioning, such as non-cluster PCDH can promote the cell's motility. So we find in the mandible pupfish, which contains 14 cluster PCDH genes. This number is similar to other fish. such as zebrafish and Madaka and Fugu. But the cluster PCD genes in the monoidal, which lacks the beta subcluster. It's interesting. We find the non-cluster PCD genes and synthesis was expanded. You can say, such as the number of PCDH. There are 3 PCDH genes in synthesis compared with only 1 in other fishes. In mammals, the PCDH genes play an important role in the embryonic development. If decrease the expression of PCDH genes, the implication site will significantly decrease. So, the higher copy number of PCDH genes in synthesis suggests that it may play a role in creating the implication sites in the male brown pouch. And our result also shows that some of these genes have expressions during the pregnancy. So we can say these genes maybe play an important role in the synthesis pregnancy. At last, we focus on the signature of the genetic comparative with placental mammals. A common feature of synthesis is the presence of the placental-like structure to which the embryonic attach. Previous study is shown that the bone pouch is functionally equivalent to the mammal's utero, and this represents an interesting instance of adaptive convergent evolution. To explore if there are the signature of genomic convergent, we used we applied CSS method to find some genes if they undergone some convergent evolution. Genome-wide comparison identified 100 antigens which with identical substitution in synthesis and placental mammals. In this method, we can find at least 3 members of synthesis and 3 members mammals have the identical substitution. We can say that they undergone the convergent evolution. Most of genes have at least more than 7 species have the same substitution. Association of some of these genes with placenta and embryonic development in mammals suggest a role for them in synthesis pregnancy, such as ASK1. These genes have 2 site substitution, and ASK1 genes play a role in placental angiogenesis, which is important for the placental development. And most of the genes have high expression in the bone pouch. In summary, first, we find the molecular evolutionary rate for synthesis is higher than that of other teleosts, and the loss of but only 1 PQ-SCP genes in synthesis suggests a role for the lost genes in dating and metabolites. anomaly formation, and TPS4 has a role in the paragraphin formation, and where our results suggest that the loss of paragraphin in syngnathids may be related to the loss of TPS4 genes. And at last, genomic-wide comparison revealed that the signature of convergent in syngnathids and placental mammals. And we find 118 genes which show the convergent evolution. And these genes play an important role in the placenta and embryonic development. We thank Professor Winky and Professor Mazer and Professor Huang for their help in my project. And thanks very much for the members in our group. And thanks these foundations. Thank you very much.
Okay, thank you, Dr. Zhang, for her talk. And now we are taking one question probably because of time limitation. Are there any questions here in the group? Raise your hands, please. Any students